Right, so I'm going to talk about our efforts on developing different approaches for promoter prediction. So my lab mainly focuses on well, transcription, translation, regulation in different microorganisms. But as simply put, we have fundamental discoveries that we're trying to understand how these processes operate. And in terms of more machine learning computation approaches, we're trying to control as much as we can understand what these regulations do involve. In terms of putting the state, one of the limitations that we currently have is that the genome sequence space is growing immensely. But when we do want to understand what is in these genomes and what functional role all these different elements do play, we are good at identifying the coding sequences, but anything beyond that one is really getting it's what really very well recovered covered so far, and kind of exemplify that. E. coli genome was published in '97. It's soon going to be 30 years. So let's say it has about 4,000 genes, and only 65% has been of the genes that have a mapped regulatory mapping. So still quite a bit of way to go in terms of moving from a static look at what is in a genome to get an understanding of what the functional role these things do play. And of course, in that, the promoters play a significant role. And why that matters? Well, there can be many examples, but one of the things that it took our attention lately was the study by Akos in George Church lab, so that they have found out that when they do synonymous recoding in E. coli, and this is in 57 and 61, there are more than 10,000 of cryptic promoters occurring. So yeah, we have the power to, create minimal genomes. We can assemble them. We can, remove one of the stop codons. But the consequences of is that, we mess up with the transcriptome dramatically. On top of that, there has been impressive developments now using generative AI for genome design. So that was lately from ARC Institute was published EVO1. And now in bioRxiv, the EVO2 is also available. So they have a 40 billion parameter model trained on any genome sequence ever published. And using that, they could also design de novo bacteriophages. I think they designed 50. One of them was functional. So generative AI has immense potential in terms of understanding static knowledge, if you will, on a primary sequence level. But our understanding, and that means functional understanding of the DNA and its role, it's missing. And this has also been exemplified by one of the recent efforts. It's the BioTOCA and BioFM framework. In this study, what they have shown that by keeping this primary sequence as the base data, there's like practical genome sequences, but not extending the genome size. However, by increasing the functional annotation of it, things like, introns and exons and, where the transcription starts and end, the coding sequences and etc., they could show that this leads to improved functional prediction using these models. So this is a kind of simplified graph showing that the same data size, no more extra genomes added. It's just that giving the data size with extra functional annotation using the BioFM network, a framework they've developed, they could show that actually they could improve the performance of prediction. So this brings us one important aspect. Yes, we have this immense capacity to sequence anything that we could get a hold on, but our understanding of the functional role and hence the functional annotation is not catching up equally, and that leads to a gap that yes, we can see DNA sequences, but that gives us a poor in terms of knowing predictive power of the function. So how do we contribute to this situation? So in my group, we have developed a massively parallelized in vitro transcription protocol. It involves generation of large number of artificial sequences. So in this particular case, we have designed 6.2 million DNA sequences, and then we screened them through an IVT protocol. In this particular case, we have been working with 3 different microorganisms, Bacillus subtilis, E. coli, and Pseudomonas putida. So we have all 5 sigma factors heterologously produced from Pseudomonas and from E. coli as well, and we have 7 from Bacillus subtilis. So in total, we have 17 sigma factors. And we also have the corresponding core RNA polymerase from these organisms. And by using the 6.2 million DNA sequences, we run in vitro transcription reactions where we add 1 sigma factor and the corresponding core RNA polymerase. And then we run this massive parallel experiments. And in each experiment, we collect the RNA and then we sequence it. And there we use 2 approach. We do RNA sequencing just to get the quantities, but we also do explicitly determine the transcription start site by doing a method called enriched 5' cDNA sequencing. So with that, we have generated so far not been reported data size that consists of 6.2 million DNA sequences. Given the 17 sigma factors, we have mapped about 4 million functional RNAs. And with the TSS method I mentioned, we know of the 4 million, 3.5 million transcription start sites. And this allows us to now not just statically knowing, where the promoters are, it also allows us to generate the genotype-phenotype mapping. What we have done so far, if we were to giving any sequence, the models are able to predict whether the sequence is functional or not functional. And this is an important parameter to consider. Non-functional means when we generate these large data sets, we just don't only have sequences that do work, we also generate a quite large data set that is consisting of non-functional sequences. Why is that important? There is a phenomenon called negative data set in model developments, and the richer the negative data set is, the better the models do learn. Hence, this approach, plus in generating functional sequences, but generating large number of non-functional sequences makes the model learn much better. And with that, we're also able to, giving the sequences, we can not just predict if there is a functional sequence or not, but we are also able to predict to some extent, in terms of expression level, we are taking a bit simple approach here, saying high, medium, and low. And then one of the other aspect of this promoter prediction is that we will be going, not simply going, knowing, whether you have a functional promoter or not, but we are also able to tell with certain predictive accuracy which sigma factor actually happens to be present in the given sequence. And this is just a demonstration of 7 sigma factors from Bacillus subtilis, and this has been mapped on the Bacillus genome. So how do we go about it? What we want to do now is, so the first study is nearing completion. Instead of just simply annotating coding sequences, we want to also be able to annotate the regulatory sequences in genomes. Currently, we are operating within the bacterial domain. And this what has a consequences on rational synthetic genomic design. One thing that, as I have exemplified now from Akos's work, is that this will hopefully allow us to avoid occurrence of cryptic promoters when we design minimal genomes, or we want to do synonymous encoding, for instance. And with that, we will be able to not just predict promoter activity, it will be maybe to some extent on the strength of expression and plus which sigma factor might be applying on this particular sequence. And one outcome of that will be that we will be also be able to design promoter sequences with some predictability. Now, coming back to this consortium, of course, sort of for this community, how do we go about using our approaches for JCVs? And so here is maybe one more step back. I presented you so far E. coli, Bacillus, and Pseudomonas. These organisms have many sigma factors, as you have seen in the previous one. But JCV-ISN is a bit of special. It is a single sigma factor. So then the question is, how does it coordinate the entire genome with a single sigma factor? This gives us a good test bed opportunities. We can maybe try to look for, what are the other features? And I think John last nicely put it, what are the time signatures? How would an organism decide, giving with one sigma factor, which gene should be expressed 1 now and number X maybe in 2 hours period? And also with this having the the mother strain, the SIN1 and SIN3 and different versions, what type of reorganizations or what consequences of the reorganizations that we do see? And these type of, or not exactly, but these are some of the goals that we would like to pursue. So we have gotten hold on to some data from John. We're thankful for that. But we found out that we have to run some experiments ourselves. And here is just a simple screenshot of the different tools that we have developed and what it allows us to actually, we can look at the genome and we can tokenize the data, but then we can ask the models, can you show us, where do you think a transcription may start, giving what we have trained the models on? And this gave us some inputs. Of course, we want to build these further. And these were type of approaches that we would like to address using JCVI, SIN1 and 3A and 3B. Yeah, so that was a really brief overview of the different things that we have been doing. I think here are two important people that I would like to mention. One is Isa, who has generated this really large data set, and Erlen is currently using this data set to train the deep learning models. And we have various partners, University of Bielefeld from Czech Academy of Sciences, Libor, who provided us with all the core RNA polymerase and sigma factors of Bacillus, for instance. Different innovation funds and granting agencies. Yeah, with that, I'll thank you and I'm open for questions.
Fabulous, Rahmi. So questions, I see applause. Do I see hands raised for questions? Tae-suk or James?
Sure. Great talk and good to see you again. I just wondering how generalizable your approach is, also how your performance compared to the Howard Salis promoter prediction algorithm.
Well, let's start with the second one. I mean, I'm not updated on the current status of the Salis approach, but if I'm not wrong, they have E. coli sigma 70 and a few others as the base. And the issue is that, or the limitation, maybe not much of an issue, is that those are based on designed, I don't remember the number, some 10,000s of promoters. So one of the limitations of developing machine learning was, and I think that we are seeing it more and more here and there, you can generate the data and you can train a model based on data, and the model performs beautifully, giving the spheres or limitations of the data. As soon as you step aside, it falls apart. And that's what we have seen. There are 10 million promoters from yeast. It works great, but it's a single promoter structure, and they use the randomization to go abroad. So that introduces a limitation. And similarly in Salis work, I think again, I do not know the details, but if the data comes from a very limited condition, in this case, sigma factor single host, you can't generalize it. So that's what we're trying to avoid, with going away from a single host to multiple host, but that means it says it for itself, it's limited. Bacillus has 19 sigma factor, we will be able to only use the 7, et cetera, et cetera. So we don't cover the entire sequence space, but by going beyond it, we are hopefully able to generalize a bit more. And can you come with your first question once more, please?
How generalizable your approach is, because you only also touch 3 different species, and then going into the new species, including the
so generalizability
JCVIs.
is so that we're improving the state. It's not generalizable enough to cover the entire spectrum of transcription regulation. We have different measures to tackle that. So for instance, one thing that we are doing is now, we can't do this for every organism. So how can we go about it? So what we came up is that we will do transcriptional factor embeddings and take those into our models, because the 3D structure of a transcription factor should be able to, and especially with the AlphaFold 3 docking model available, we want to take those information to kind of get some educated guess on giving the transcription factor, where these might bind. This will hopefully be able to generalize. Currently, you can't go beyond more than what we have, but thanks to the evolutionary concern transcription processes, kind of the core sigma factors we cover is conserved across. So that kind of gives a bit of room, but we will soon know. We are currently limited to only these hosts, but we're going to apply soon to different organisms that we have taken. We're also collecting RNA sequence data so we can kind of meet validations and etc. to go beyond single host, single sigma factor to more generalizable approaches.
Okay, thank you.
James?
Thanks. Very exciting work, Rahmi. It's great to see you. I think a related question, mycoplasmas have a low GC content. For example, the GC content of JCB isin 3A is only 24%. And I'm curious how this information, if this is somehow implicit in the model.
Not in the model, it's not implicit at all. It just sees it's, I mean, I can say it in this way. The 6 million data we have, it goes from, what, 20-some% to 70-some% GC content. So unintentionally it represented, but of course the functional ones are only ones that are working off these 17, right? So it's not a parameter in the design, but it's there in terms of sequence and function relationship. And going back to the SIN, what we want to do is actually we want to embed different features, and comes back to actually really nice the first presentation. It is not just about, whether you're finding a motif or not, but, how the nucleoid structure is, or where is the promoter in the given genome, how the folding is. So we want to take multiple parameters when we do this work. And of course, depending on the funding, we haven't started yet. And that's kind of goes under the category of, just going beyond the canonical motifs. Can we take these different parameters into picture as well? Of course, we will not go as wild as, considering this charge and all that. The model will have limitation, but at least we want to go beyond the canonical motif understanding. Can we go say more than just -10, -35 boxes?